Do subscribe to Ekeeda channel and press bell icon to get updates about latest engineering, HSC and IIT JEE main and advanced videos.
Hello students, we are doing chapter Genetic Engineering and Genomics. Today we'll be studying about how insulin was made in lab. We all have seen insulin bottles, or people who are diabetic take insulin injections. Do you all know that this insulin injection was made by technique called as genetic engineering? Yes, it was made in lab by using the technique genetic engineering. And today we are going to study what is this genetic engineering and how insulin was made. Hello students, we are doing chapter Genetic Engineering and Genomics. In that, we are studying today about insulin. How insulin was made in lab. We all have seen people taking, diabetic patients basically taking insulin injections. Do you all know that insulin was formed in lab and it was formed by the process called as genetic engineering? Yes students, this was the process that was used to make insulin inside labs. So now, long back ago when insulin injections were not available, still people had diabetes, so what was done? That time what was done was horses and pigs were slaughtered or killed, and from their intestines or pancreas insulin hormone was removed, which was available in small quantities, and this small quantity insulin hormone was given to diabetic patients so that they could survive. Now it was not at all feasible to kill so many animals and give insulin injections to so less patients. So what scientists thought, that let us make insulin inside lab. While making insulin inside lab, scientists had three main aims or three main parameters which they wanted to fulfill. The first parameter which they wanted to fulfill is the amount of insulin made in lab has to be larger quantity, since many people had diabetes. The second parameter was the insulin that was made inside lab has to be similar to the insulin present inside human beings, because human body has a tendency to reject or not to accept anything that is foreign. Third thing, cost effective. The insulin made in lab has to be cost effective. It should be feasible and easily available to everything. So now, keeping all these parameters in mind, they started to make insulin inside lab. What they did, they took human pancreas, okay, obviously from a dead individual. They took human pancreas, and from this human pancreas they isolated insulin gene from the beta cells of human pancreas. Now they isolated the gene that produces insulin hormone, basically the beta cell genes, and now this beta cell gene was kept inside a vector or a carrier. Okay, now what is the function of a vector or a carrier? The carrier or a vector is nothing but a vehicle that is used to carry one thing from another place or one place from to another place. So now this carrier along with that insulin gene, which you can call vector, carrier or vehicle, was introduced into E. coli or Escherichia coli. We all know the duplication time of Escherichia coli is 15 to 10 minutes. So when Escherichia coli or E. coli will multiply if it is given proper food, nutrition, this insulin gene along with that vector or vehicle kept inside the E. coli will also multiply, and this way a lot of copies of insulin gene will be made along with the vector, and finally we will break open the E. coli and obtain insulin hormone by certain more processes. You all will say that how is it possible an E. coli will accept something which is foreign? Usually your body has a tendency to reject anything that is foreign, and even that applies to E. coli. Correct. First thing, the plasmid, the vector or the vehicle that is obtained or used to introduce the gene is obtained initially from E. coli. It could be PUC19 or PBR322. Second thing, our genome and E. coli genome is 99% similar. So when you introduce a plasmid that is obtained from E. coli inside E. coli, it will readily take it up and make copies of it. During this technique, or while doing this technique, you all must have noticed that this technique is nothing but genetic engineering. When I say genetic engineering, it means manipulating the already present or the genes or chromosomes which are available in nature for our own individual purposes, for our own benefits. Have you seen in genetic engineering, in this technique we used human pancreas, human gene, and then that human gene was kept into E. coli, two different organisms, eukaryote and prokaryote. So this technique is nothing but genetic engineering, where you combine two different, entirely different entities or properties. This technique was also called as rDNA technology or recombinant DNA technology. When I say recombinant or recombining, it means mixing. You all must have noticed that we recombine a plasmid with insulin gene and kept it inside an E. coli. Now, when E. coli will multiply, it will also make copies of plasmid as well as the insulin gene, thinking this all belongs to its own genome system, or thinking they all belong to its own environment. Basically, the recombination of gene happens, due to which insulin is produced. So now let us see how insulin is made in lab. Let us see how insulin was made inside lab, or insulin production in lab. We took human pancreas, from that insulin gene was isolated. Now this insulin gene was kept inside a vector or a vehicle or a plasmid. Now this insulin gene along with that vector or vehicle was introduced into an E. coli. This E. coli had the plasmid or the vector or the vehicle along with that insulin gene. So when E. coli will replicate, copies of insulin gene were made. Then we break open E. coli to obtain insulin. After certain synthesis processes, we get the final insulin product. Basically, rDNA technology or recombinant DNA technology was the technique by which insulin was made in lab. Production of insulin using genetically modified E. coli was first done in Canada using vehicle, vector or plasmid PBR 322. Thank you.